Now I make around you all the numbers. All those super heavy yellow rubber heavy birds.
Might be Kevin joining too.
Yes.
The tallest person sits in the front row.
Tracy, Derek, Todd Morris from Fisheries and Oceans Canada, and Nick Sard, who's a faculty member and molecular ecologist, and helped train our lab when he was a postdoc at Michigan State. And now he's at SUNY Oswego in New York.
Are there people with these things?
Yeah, there's a group with Nichelle in her group too. She's on the committee.
It is. That's why I was in the.
Hey, Alec.
I got a heating pad.
Yeah, you plug them in.
That's a thought. I might go get my coat.
I invited everybody and reminded there's coffee and donuts.
The reminder of food is
Always a good hook. Beyond the excitement of Nichelle.
Sorry
Hey,
to
Derek.
have coffee for y'all, but we can go have alcohol after.
No. There's no.
Again, University. That's where my
Well, the committee will gather closer.
They'll gather
Afterwards.
Yeah.
That's Nick.
Hi, Courtney. Hi.
Nick and Wave.
Hi, everyone.
How can you tell there's a movie from here?
They're automatically muted.
I can't, you're right. It doesn't stay in the little corner.
No.
It would be a little something in that, right?
Yeah, but not on, that. All those be helping me.
Yeah, should the participant list.
Okay, here we go. We can see the chat.
Going after this.
Anybody? Oh, it looks like there's stuff happening in the chat though.
Samples are collected, no more. Just gotta have it.
It's not nice.
Is somebody saying something in the chat?
I'm trying to figure. I don't know how to use WebEx.
For right, the little.
Other right?
Right.
It's right under the participant list if you're looking at like the full screen, Dave. Sometimes when you're presenting, it shows up in like the WebEx app box.
Nichelle knows everything. Oh, come on. About doing
We're going to get started in a minute or two. For those in the room, you're welcome to grab coffee and donuts or fruit if you feel so inclined. Yeah, you can close the door, but you don't have to latch it.
Yeah, it's a really nice big conference table in that room.
Someone have it.
Eight.
No statement.
Okay, Nichelle, and if you want to mute yourself on your end so we don't get extra feedback.
Yep, can do.
Just for now, and then when I'm done, we'll switch sharing over to you, and then we'll do kind of the same with muting ourselves over here and just having our volume on. All right, so it's my pleasure to welcome everyone. We have a pretty big group joining us from all around the continent. I haven't seen anybody overseas yet, but that's possible too. for Nichelle VanTassel's PhD dissertation seminar. It's titled Conservation, Genomics, and Evolution of Lamp Mussels and Kidney Shells. I'm going to give a brief introduction about Nichelle, and then Nichelle's going to take over and tell us about her dissertation. So Nichelle comes to us from Florida. She is raised in Port Charlotte, Florida, on the Gulf Coast of Florida. Funny thing, my parents actually spent many winters after their retirement in Port Charlotte, and we know very similar places and been to the same beaches and all those things. There was lots of small world kind of stories when Nichelle was first starting. Nichelle has her bachelor's and her master's degree from the University of West Florida. Um, After her bachelor's, she spent some time in an internship program at the Moat Aquarium in Sarasota, Florida, and then returned for a master's back at University of West Florida in Pensacola with her master's thesis titled Characterization of Fish Communities in the Northwest Florida Coastal Dune Lakes Using Environmental DNA Metabarcoding. So she actually came quite well trained already in molecular methods and aquatic methods, although we switched her into freshwater systems during her PhD work. And her master's supervisor was Dr. Alexis Janisic. Um, and she started her PhD here in the summer of 2018, and she worked diligently with collecting a lot of data collecting a lot of specimens across the Midwest, and you'll hear about that. And then COVID happened, and we had lots of delays. Nichelle got her genomic libraries prepped on the day before all the lockdowns started. So she submitted them to the Michigan State University's genomics facility for sequencing, and they emailed us and said, yeah, we're shutting down because of the lockdown, and we'll let you know when things reopen. And they sat in their freezer for about 3 months until they were ready to reopen. And in June, they got sequenced, and she had lots of data. But there was a long waiting period in there, which was, but we persevered. And something about Nichelle and all of our students who've had to deal with all these COVID issues is it's a reflection of how strong they are as people and how strong they are as scientists as well. So this kind of word cloud that I'm showing here is actually a word cloud that was created from Nichelle's Google Scholar. So her Google Scholar page, there's a shiny app that puts a script in and creates this based on your titles and abstracts and so on that are available on Google Scholar. And you're going to hear a lot of these words repeated during Nichelle's presentation. So Nichelle is defined by her core values that are centered around diversity and inclusion, and it helps make her an outstanding aquatic biologist, a genomicist, a bioinformaticist, a coder. She's an amazing scientific communicator, and you'll see this, I think, reflected in her presentation today. She's also a leader, a mentor, a teacher. She was a TA for Bio 111, which is our Foundations in Diversity and Evolution class for a couple semesters here during the height of COVID, which was not an easy task either. And she's an amazing colleague as well. I'm proud of all of her achievements, and I think we all are really proud of all her achievements and what lies ahead in her career. And that career has already kind of started for her. She, in May of 2022, she left CMU with all but dissertation completed, and began a technician slash postdoc position at the Smithsonian National Zoo, working with Dr. Robert Fleischer on a variety of bird-related conservation genomics questions using their amazing facility at the National Zoo right in Washington, D.C. So it's a pretty cool place to work and thrive as a scientist. Nichelle's going to show this slide as well with her acknowledgments. I would like to briefly introduce her committee. So Dr. Tracy Galarowitz is a faculty member in the biology department and Associate Dean of the College of Science and Engineering here at CMU. Derek Learman, who's here in the room, is in the biology department, a faculty member in our department. Nick Sard is joining us online. Nick is an assistant professor at the State University of New York in Oswego on the shores of Lake Ontario. Nick started 3 years ago at SUNY?
2018. So
Yeah. Okay,
yeah,
5 years ago
yeah.
already. But
Time
Nick,
flies.
Nick trained our lab in some of the genomic library preps that we've been doing, and has been an invaluable resource for a couple of graduate students now, and has been a great mentor for Nichelle as well, and guiding her through some of her dissertation work. And finally, we have Todd Morris, who is a research scientist with Fisheries and Oceans Canada. Todd and I have been longtime colleagues, and we're excited to have Todd joining us and continuing to collaborate on some really exciting work. Todd was also instrumental in helping to secure funding for this project. We were part of a assortment of Canadian universities working with the Canadian Freshwater Species at Risk Research Network that was funding from Fisheries and Oceans Canada. And we're the only American university that was involved in that, but we're working with Canadian questions and Canadian species at risk a lot in our lab. Funding for Nichelle's project, in addition to the prime source, which was with Fisheries and Oceans Canada, also came from a grant from the CMU Faculty Research and Creative Endeavors Committee, and she also had stipend support from the College of Science and Engineering in the Ecology, Earth and Ecosystem Science PhD program. And the National Zoo has also allowed her to have time to continue writing up her dissertation in the last year and a half that she's been there. So with that, Nichelle, I'm going to have you take over the screen. I'm going to mute myself. Turn up our volume. Can you check to see if we're hearing you? You're still muted.
Okay, can you hear me now? Okay, I think that was a thumbs up.
That's a thumbs up. I can hear you. You're good.
Awesome, thanks. His hand was above the screen, so I couldn't tell. Thanks everyone for joining us early on a Friday morning, and thanks Dave so much for the generous introduction. I'll give you an outline of what we're going to go over today. I'll start with the introduction to freshwater mussels and conservation genomics. And then I'll walk you through each of my dissertation chapters, starting with the broadest topic of phylogenomics of an entire genus. Then we'll narrow down into population genomics of one species. And then 2 chapters applying conservation genomics to look at genetic diversity of wild versus captive reared freshwater mussels using 2 kinds of molecular markers. I'll then summarize the cumulative impact of this work, and then I'll tell you a little bit about what I've been doing applying my skills at the Smithsonian's National Zoo, Conservation Biology Institute, Center for Conservation Genomics. So freshwater mussels are bivalves belonging to the family Unionidae, and they are often referred to simply as the Unionids. Unionids have a unique parasitic larval stage where after fertilization, females will release their larvae called glochidia, which will then attach to a vertebrate host. And in most cases, this is a fish. And the mussels have adapted or evolved several kinds of host attraction methods. And in this case, in the photo, you're seeing a lamp mussel, which has a mimic lure. So her mantle tissue is made to look like a fish, and she can articulate this to look like a swimming fish. And then a fish will come along and take a bite of that mantle tissue and get a face full of those larvae. The larvae will attach to the gills and encyst until juveniles are transformed and fall off into the sediment. And unionids, especially when they're in biodiverse populations, provide essential ecosystem services. So they're filtering water, which can remove pollutants. They're cycling nutrients. They provide a food source for other organisms in the habitat. And they also play a role in the cultural practices of indigenous people. And despite those ecosystem services and their importance, more than 70% of unionid species are imperiled, with their decline being linked to things like mass harvesting for buttons in the 1800s, habitat modification, especially relating to the building and usage of dams, pollution and runoff into rivers, and invasive species, particularly biofouling by zebra and quagga mussels in the Great Lakes. And due to their importance and imperilment status, there are a lot of restoration plans in place to help restore the mussel populations and their habitat. So this is often done by doing surveys and population monitoring, like you can see in this photo. Restoring those habitats, so things like removing dams where they're not needed anymore, and doing propagation, augmentation, reintroduction, translocations, and introductions, and I'll call these party activities. And then also the investigation of genetics and genomics. And the field of conservation genomics is focused on a variety of topics, from broadly understanding species biology and evolutionary genomics to taxonomic uncertainties, and then more specific things like population structure, fragmentation, introgression, outbreeding, and really applied topics like forensics. And the field really focuses in on small populations where the loss of genetic diversity, mutational accumulation, and inbreeding leads to decreased fitness, ultimately sending species towards imperilment and the extinction vortex. This is where conservation-minded people get involved to think about genetic management, to identify management units, start propagation programs, do other party activities, and identify adaptations to captivity. And my dissertation work focuses on these topics to understand how genomics can inform freshwater mussel conservation. So before I move into the chapters, I want to make a quick note on the molecular markers I used. So I used traditional microsatellite markers, which are short tandem repeats in the genome, and we can genotype individuals by visualizing the length of those repeats. And I'll refer to these markers as microsats moving forward. I also used more contemporary marker type called single nucleotide polymorphisms, or SNPs. And these are mutations or variations at a single base position. And using next generation sequencing methods, we can identify thousands of SNPs and genotype many individuals at once. And to produce the SNP datasets that I'll talk about today, we used a method called RAD-seq, or restriction site associated DNA sequencing. And specifically, I used the BEST protocol as described by Ali et al. In 2016. So now we can move into the chapters. And the first chapter is RAD-seq, derived SNPs reveal cryptic lineages in the freshwater mussel genus Tychobranchus. Currently, there are 6 recognized species in the genus: the kidney shell, P. fasciolaris, the Washita kidney shell, P. occidentalis, the fluted kidney shell, P. subtentis, the rayed kidney shell, P. forminianus, which was elevated from synonymy with the triangular kidney shell, P. greeni, based on shell morphology. And lastly, the southern kidney shell, P. jonesi. And kidney shells specialize on darters and other benthic fish for their parasitic larvae, and they package their larvae into these things called conglutinates. So they're little packages full of larvae, and they're made to look like prey items to attract their host fish. So fasciolaris and occidentalis both mimic fish fry, and you can see here this is fasciolaris, This is occidentalis. And the bottom picture, you can see where the conglutinate has broken open and all of those larvae are spilling out. Subtentis mimics black fly larvae. Jonesii mimics insects, eggs, and larvae. And greeneye mimics fish eggs. Presumably Foraminianus also mimics fish eggs, but conglutinates for greeneye versus Foraminianus have not been differentiated yet. And here I'll show you a short video. So this is a female subtentis, and you'll see the release of those conglutinates on the glass there. And then you'll see a darter try to eat one. Now you see it just spit it back out. And while it was in its mouth, it was able to get all of those glochidia released and attached to the gills. Entychobranchus is an ideal genus to focus on phylogenomics of freshwater mussels because they have a widespread distribution. And there's well-documented literature on the phylogeography of their host fish. And since they depend on their host fish for dispersal, patterns in the host fish can help us elucidate patterns in the unionids. Their evolution has also been influenced by recent forces and more ancient forces. And in aquatic species, we see phylogenetic breaks across major watersheds due to historic isolation. And then we see fragmentation within watersheds based on more recent barriers to gene flow from things like dams, waterfalls, and glacial events. And during the Pleistocene glaciation, we know that organisms moved south to refugia. And then after the glaciers receded, organisms were able to colonize north of the glacial maximum, and lots of genetic change happened rapidly during that time. And we can see evidence of these gene flow patterns in the genetic data. Additionally, 4 of the 6 currently recognized species are federally endangered in the United States, with a 5th being endangered in Canada. And there's also a published mitochondrial DNA-based phylogeny, which confirmed the genus as a monophyly and provided some hypotheses. So the main objectives here were to determine the number of species in the genus Tychobranchus, describe the evolutionary relationships within the genus, and then compare our results to the mitochondrial DNA-based phylogeny by Rowe in 2013. So we were able to collect and source a bunch of Tychobranchus from across the range with the help of lots of amazing mussel folks. And after library prep and bioinformatic processing, we were left with 118 Fasciolaris from the Great Lakes, the Wabash, the Ohio, and the Tennessee rivers. 4 Foraminianus from the Cahaba, 2 Jonesii from the Choctawhatchee, 68 occidentalis, and this includes 2 regions that I'll talk about. So the Ozark region will be the northern 2 populations, which are the White St. Francis rivers and the Little Red River. And then the Washita region encompasses the Red River and Washita River in the south. We ended up with 10 subtentis from the Cumberland and the Tennessee River. And unfortunately, none of the few greeneye samples we were able to obtain made it through quality control, so they will not be included in these analyses. And here we have a consensus tree made with 9,333 SNPs. This tree was made using IQ tree, and node values are bootstrap supports based on 1,000 iterations. And I'll walk you through the tree from right to left, going from more ancestral to more derived lineages. The text in black will reflect the current taxonomy, and then colors will reflect collection drainage. So let's start with subtentis. So we see that subtentis comes out sister to the rest of the genus. Oh, and note that as highlighted on the tree, there will be circles on the map that will orient you to where we are in the geography. So subtentis comes out sister to the rest of the genus with 2 reciprocally monophyletic clades associated with the Tennessee and the Cumberland. And note the bootstrap supports are really high, with each of the lineages that I'll highlight today having 95% or higher bootstrap supports. Next, we have Jonesii and the Choctawhatchee, which comes out sister to Foraminianus and the Cahaba. And then we have 4 lineages in Occidentalis. We have 2 associated with the Washita. So those are the 2 southern populations in the red in the Washita River drainages, and 2 in the Ozarks, with 1 being the White St. Francis and 1 in the Little Red. And note that these 2 Ozark lineages are paraphyletic with our Fasciolaris lineage. And Fasciolaris does seem to have 1 apparent lineage, with the topology indicating a path of postglacial colonization from the Ohio through the Wabash into the Great Lakes. And when we look at a DPC analysis, which takes principal components and forces them into groups based on the best fit number of groups, we see that the best fit number of clusters for Fasciolaris is 2. So this plot is showing you the x-axis of a coordinate plot, and then the y-axis is density of points at that place on the axis. And so we see here 2 clusters, but we see considerable overlap in the center of this axis. And the axis is pretty small, ranging from -6 to 4. And then in Occidentalis, we see 4 groups is the best fit. And the axis here is huge, going from, I think, -8 to the 16th, all the way up to 4 to the 16th. So this is indicating that these 4 Occidentalis clusters are much more different from one another than the 2 clusters in Fasciolaris. So this is more evidence that these are distinct lineages or species. Oh yeah, and that's the southern 2. Okay, and then subtentis, again, we have 2 clear clusters here, a more moderate axis, but no overlap. So this again is indicating 2 clusters or 2 lineages in subtentis associated with the Tennessee and the Cumberland. So to test whether these lineages we see on the tree and in the cluster analyses are in fact distinct species and not just patterns of structure based on geography, I did a species delimitation analysis in Beast 2, where I tested 5 models of speciation. A, the current taxonomy, with 5 species, because green eye is not included. B is the same as A, but with a split in the 2 regions of Occidentalis. So 1 species in the Ozark, 1 species in the Washita. Model C would be a drainage level split in Occidentalis, so 4 species within the Occidentalis clade associated with rivers. Model D is the same as Model B, but with an additional split in subtentis. So 2 species of subtentis, 2 species of Occidentalis. And then Model E, a drainage level split in Occidentalis and a drainage level split in subtentis. So 4 Occidentalis and 2 subtentis in this model. And what we find is that Model E is the best fit. So based on maximum likelihood estimation, this model ranks number 1. And then the negative value in the Bayes factor, which is this BF value, indicates that the alternate model is better than the original model. And then this value being so large at 46,000, based on conventions, is decisive evidence for this model being the best fit. So 9 species in the genus Tychorhynchus rather than the 6 or 5 without green eye. So then if we compare these findings to the mitochondrial-based DNA phylogeny of Rowe, pictured on the right, we both find the genus to be monophyletic. We both find subtentis sister to the rest of the genus. And we both find Occidentalis, and specifically those Ozark lineages, paraphyletic with Fasciolaris. There are some differences though, notably the SNP-based tree finds Foraminianus sister to Jonesi rather than sister to the Fasciolaris occidentalis clade. And again, we didn't have green eye included in our study, though Rowe treated Foraminianus as green eye in his study. So we would expect, if we had green eye, for them to come out with Foraminianus. Lastly, Rowe identified 2 lineages in Occidentalis, one in the Ozark and one in the Washita. With SNPs, we identified 4 lineages, 2 within each of those regional clades. The implications of these findings are that we would recommend that new taxonomy be described, with morphological assessments needing to be done. And the cryptic lineages in the Occidentalis clade would mean that it's not so secure. So currently it's the only species of Tychobranchus without any protections. And if there are 4 lineages here, that means there are 4 narrow range endemics instead of 1 more widely distributed species. So that would take additional assessments. And unfortunately, we were unable to resolve the relationship between greeneye and Foraminianus. So that's still unresolved and will need further study. Okay, so now we're going to move into chapter 2 and narrow in on Tychobranchus fasciolaris and discuss how rad-seq generated SNPs resolve patterns of genetic diversity and structure of the freshwater mussel Tychobranchus fasciolaris in glaciated and unglaciated regions of North America. So the main objectives are to examine genetic diversity of fasciolaris across its range and identify management units for conservation. And this is important because while the kidney shell is secure in most of its United States range, it is endangered in Canada and of special concern at the northern extent of its US range in Michigan. So cluster and genetic analyses with 9,978 SNPs reveal 2 genetic groups. Again, the top is a DEPC, like I showed you in the first chapter, and then the middle plot is an admixture analysis showing you ancestry coefficient on the y-axis. Each bar is an individual, and they are oriented based on geography, with Great Lakes on the left to Tennessee on the right. The bottom plot is a structure analysis, which has the same orientation as the middle plot, but the y-axis here is membership probability. And what we see with all of these is 2 clusters, one associated with the Ohio, Tennessee, one associated with the Great Lakes. And then we see a nice pattern that shows us the path of colonization in the admixture plot, especially. So we see this step down pattern with mixture starting in the Wabash population down into the Great Lakes. And when we put this back on a map, this path makes a lot of sense. So I added a dashed blue line here to approximate where the edge of the ice sheet would have been. And you can see here where the path was through the Wabash into the Great Lakes. And we see more evidence of this path with clear isolation by distance patterning, where the more geographically distant individuals are, the more genetically distant they are as well. And when we look at genetic diversity metrics, we see lower heterozygosity, nucleotide diversity, and fixation between populations in the Great Lakes compared to populations in the Ohio-Tennessee cluster. And this is indicative of a founder effect from post-glacial colonization. The global fixation index between the Great Lakes and the Ohio is 0.194, which indicates some isolation between the 2 groups. And when we look at a fine rod structure plot, this is a co-ancestry matrix, and I'll orient you to this, so don't get too worried. The color ramp here is showing lowest relative co-ancestry with yellow values and highest with dark blue, black values. Each square is a pairwise comparison. Each row and column is an individual. And then the tree at the top is the result of a cluster analysis, which is reflected by labels on the bottom of the x-axis. The labels on the y-axis will orient you to the 2 genetic groups indicated by previous analyses. And so when we look along the diagonal line, we see these small pink and purple boxes indicating substructure by river, with a few notable exceptions. We see here this big chunk of the Ohio where they're all very similar to one another. There's not a lot of structure in this section. So there's a lot of gene flow between these rivers in the Ohio River drainage. And here between the Sydenham and the Bell, there's higher gene flow than between other rivers in the Great Lakes. More evidence for the Wabash entrance of fasciolaris into the Great Lakes is shown here. So this is the Wabash populations. And when we look at co-ancestry with the Ohio on the left and the Great Lakes on the right, we see very similar values, but on average higher in the Ohio, except for this population. So this is the St. Joseph, and the Wabash population has even higher co-ancestry with St. Joseph than it does with any of the others. So this is indicating that the St. Joseph was likely the first population colonized by fasciolaris in the Great Lakes. And the implications of these findings are that the Great Lakes and Ohio, Tennessee populations of fasciolaris should be managed as separate units. And as many previous studies have found, best practices for party activities are to remain within watershed. Though based on our co-ancestry analysis, there could be an argument to augment or translocate beyond watershed where that co-ancestry is high and where high gene flow is happening, for example, between the Sydenham and the Bell. Okay, so now we're going to move into the more applied conservation genomics chapter. So this is chapter 3, genetic diversity maintained in comparison of captive propagated and wild populations of Lampsillus fasciola and Tychobrachus fasciolaris. And you can read this full study in the Canadian Journal of Fisheries and Aquatic Sciences if you'd like. And a shout out to my co-authors, Todd Morris, Christopher Wilson, and my PI, Dave Zanata. Okay, so before we move in, I want to just review some terminology surrounding captive rearing that might be helpful. So when I say dam, I'll be referring to a mom or a female used for captive rearing. Sire will be a dad. And then when I refer to their children or offspring, I'll use the term progeny or juveniles. So our main objective for this chapter was to determine if genetic diversity is maintained when we captive rear unionids. So we collected 12 gravid or pregnant dams from the Grand River and 7 gravid fasciolaris dams from the Sydenham. And then fish were inoculated with their Glochidia and mixed, and then all of those fish were kept mixed in tanks until the juveniles transformed and fell off, and then all of the juveniles were mixed together and kept until they were large enough to sample, and we were able to get 50 juveniles from each of those species. Unfortunately, we were not able to obtain genetic material for the dams in this case, and 8 microsatellites were used in this study. So no significant differences were detected between genetic diversity metrics, indicating that there was no loss of genetic diversity between the wild population and the captive reared juveniles. And when we look at genetic differentiation matrix for fasciola, we see that the progeny and the wild grand population show low differentiation and slightly higher differentiation when you compare them to another river which is known to show genetic structure. When we look at fasciolaris, we see even lower differentiation between the progeny and their wild population. And we can visualize these differences in 2D space with a principal coordinates analysis. So here in Fasciola, we do see the expected separation between the Thames River and the Grand River. And we see complete overlap between the Grand River and their captive reared juveniles. In fasciolaris, we see the same kind of pattern, complete overlap between the progeny and their wild population. So implications of these data are that captive rearing efforts can maintain genetic diversity when at least 7 to 12 dams are fertilized in the wild. The question is then how? We think this is likely due to multiple paternity, which unionids are known to do, but how many? And is it enough? How many are enough? We have lots of questions about this that we were unable to answer with just 8 microsatellites and without information from the dams. So further study is needed to understand the parental contribution and variability between dams. Okay, so now moving into the last chapter, I will present a comparative assessment of genetic diversity for captive reared Lampsillus fasciola using microsatellites and rad-seq generated SNPs. So with chapter 3 in mind, our objectives were to assess genetic diversity and structure in wild and captive reared Lampsillus fasciola, determine if genetic diversity varies between broods, and assess the male contribution to captive reared juveniles. And then we wanted to compare the patterns revealed with microsats versus SNPs. So this go around, we were much more intentional about the hatchery methods so that we could address those objectives. So we collected 12 gravid or pregnant females from the Grand River again, and then back at the hatchery, their glochidia or larvae were flushed with sterile water. And then we took a tissue sample from the dams. And then we checked viability of those glochidia, and based on density, we inoculated their host fish, the smallmouth bass. We visually checked to make sure that attachment was successful, and then the fish were kept grouped in tanks based on which dam's progeny they were inoculated with, so that we could trace the progeny back to their mom or their dam. Once the progeny transformed and fell off, they were then kept in buckets until they were about 1 centimeter in size, at which point they were collected whole. So when we look at principal components analysis based on 7 microsatellites, we see a same pattern as we did in chapter 3 with structure between the Thames River and the Grand. But we see overlap again between the wild population and both cohorts of captive reared progeny. When we get rid of the Thames River and zoom in on the Grand, we still see no differentiation between the Grand River and captive reared juveniles for either cohort using microsatellites. And with 9,918 SNPs, we see a complete overlap again, but substructure between the family groups. And what we're really seeing is each of these groups of siblings are pulling out to their dam in PC space. And when we look at that fine rad structure plot again, we have a really nice picture of the relationships between the family groups across this diagonal. So each square here is a family group, with the middle section that's all orange being the wild population. And where you see these nice dark blue and black lines are where we're comparing progeny to their dam. So this is evidence that multiple paternity is happening here. If all of these siblings were full siblings and all had the same dad or sire, we would expect these squares to be all pretty much one color or very dark shades of blue and purple. We also see similar co-ancestry across family groups compared to the wild. So looking down this column, we're comparing each family group to the wild population, and we see a similar patterning of orange and red, which is indicative that each family group similarly reflects the wild population. We also saw genetic diversity was maintained with very similar allelic richness, heterozygosity, and nucleotide diversity when comparing the family groups and comparing to the wild population. Though we did see lower FIS or inbreeding values when we compared the family groups. And this is pretty typical when you compare highly related individuals. Okay, so now let's get into the situation of multiple paternity and trying to figure out how many sires are contributing. So we did pedigree reconstruction and then calculated the number of effective breeders. So this metric is an estimate essentially of how many breeding individuals there would be in an idealized population that shows the same amount of genetic diversity. So in this table going across, we have the number of progeny genotyped, number of dams genotyped, and then the inferred number of dams and sires based on the pedigree reconstruction, and then effective number of dams, sires, and total effective number of breeders. So using the microsatellites and the cohort from chapter 3, where we don't have any information on the dams, but we know 12 were used, we had 47 progeny genotyped, and the pedigree infers 16 dams and 28 sires. So it's interesting here that the microsatellites and the pedigree overestimated the contribution of dams to these progeny. In the new cohort, we have 180 progeny, 12 dams genotyped, 12 inferred, and 67 sires inferred. When we combine the progeny from both cohorts for the analysis, we have 227 genotype progeny, 12 dams genotyped, but we know there were 24. And the pedigree in this case infers 22 dams and infer 69 sires. So really interesting here that this combined value of 22 inferred dams suggests that either dams were reused between years or very closely related dams were used in those 2 years. With the SNPs, we had 42 progeny genotyped, 5 dams genotyped, and 5 inferred dams, and 41.5 inferred sires. And the big takeaway from all these numbers is that many more sires contributed compared to the dams. And the effective number of sires, especially using SNPs, we see is an order of magnitude higher than the inferred number of sires. So this supports our prediction that multiple paternity is responsible for that maintenance of genetic diversity. And lastly, the total of effective number of breeders is pretty similar across combinations of progeny and marker type. So when we compare our findings using these 2 different markers, we see similar patterns of genetic structure using both marker types. With microsatellites, we're able to use a lot more individuals, but with SNPs, we get a lot more genetic information, even from a smaller number of samples. The microsatellites had a lower power of resolution in the parentage analysis, often unable to correctly assign progeny to their known dams, while the SNPs provided a higher resolution when determining co-ancestry and relatedness patterns. And the implications of these findings are that individual wild fertilized gravid Lampsillus fasciola can produce diverse broods, which validates current hatchery practices. But we should still be cautious when collecting dams for party activities. We should avoid reusing dams in consecutive years when possible, and tracking which dams are used and when would be really good practice. Maximizing the number of dams used would be great when possible, and considering genetic structure would be important. So we wouldn't want to release progeny captive bred from the Grand into the Thames or vice versa when we know they show genetic structure. And the cumulative impact of my dissertation work advances the knowledge on conservation, genomics, and evolution of freshwater mussels, explores the evolutionary history of the kidney shells, employs population genomics to understand the evolutionary history of Fasciolaris in context of geographic and geologic processes, and assesses genetic diversity and structure in captive reared juveniles using 2 marker types. Insights from all of these data will play roles in conservation strategies for freshwater mussels and overall contributes to bringing freshwater mussel research into the genomics era. And over the past 1.5 years, like Dave mentioned, I've been applying my skills at the Smithsonian's National Zoo's Conservation Biology Institute Center for Conservation Genomics. So from May of 2022 to May of this year, I was a research technician working with Dr. Robert Fleischer on his avian malaria research to help understand the spread and evolution of avian malaria in Hawaiian birds and beyond. I also got to do some shotgun sequencing to help identify pathogens in mammals. And then since May of this year, I've been working with Dr. Fleischer again as a postdoctoral fellow on a couple of projects that we're getting started on the population genomics of short-eared owls. So we're really interested in understanding historic and contemporary drivers of population structure in non-migratory populations, particularly in the Caribbean islands, and on honeyguide microbiomes, which are one of the few birds that are able to digest wax. And we're really interested in how and why. So if they're able to how they're able to digest wax, if microbes play a role in that, and where do they get the microbes, if so. And so with that, I'd like to thank everyone, especially funding, my dissertation committee, people from the Zanata Lab who helped with field work, lots of amazing mussel folks who helped with the hatchery work, sample acquisition, field work, and beyond, and my family and friends who were a great support over the years. With that, I'm happy to answer any questions, and you can access mine and Dave's websites to learn more about our research and to contact us using these QR codes.
All right, thank you, Nichelle. For those of you joining us online, if you have a question, please, you can use the chat or you can unmute yourself.
Looked like Shay had her hand raised.
I will try to monitor the chat as well. And anybody from the room are welcome to ask questions as well.
I have a question in the room over here, Dave. So I'll-
Okay, go ahead, Nichelle.
This is probably really dumb. How often can the day?
Okay, let me repeat it for everyone. For everyone online who didn't hear, Steve asked that how often in a dam's life can she get pregnant or become gravid? So different species have different timelines on this. So for Fasciola, I think their age of maturity is around 5-ish, and then they could potentially be gravid every year of their life after that.
Okay.
Probably not every year, but like could. Yeah. I have one more question in here. Yeah, Rob.
Microsatellite computer.
Yeah.
Those things are highly variable. Yeah.
That's a good question. I want to say it was like 8 to 12. They're pretty variable. Yes. Rob was asking how variable the microsatellites were. Yeah, the SNPs definitely outperformed the microsatellites. Yeah. Sally.
So, in the last two chapters, you talked about the conservation, mentioned that the dams were all wild fertile.
Right.
Is it possible to fertilize dams and see work? Is it because of this multiple paternity that you would have to have so many males in captivity to successfully fertilize? Like, has that been tried? Does it fail?
Yeah. Okay, so Sally asked, I mentioned that our dams were wild fertilized, and she asked, is it possible to do fertilization in a hatchery? Why isn't it done, etc. So it can be done. There is good evidence that's not a good idea, though. There's a paper that came out by Wacker et al. That looked at the difference when fertilization happens in the wild versus in hatchery, and you lose a lot of genetic diversity and effective number of breeders when you fertilize in a hatchery, which makes a lot of sense. You're reducing the number of adults who could contribute just to what you were able to collect. So if you can, having fertilization happen in the wild is much, much better.
Nichelle, there is a question on chat. Do we know if multiple paternity occurs across all species of Unionids, or is it more likely in certain species or genera?
That's a good question. I don't know if we know that. I think a lot of the multiple paternity stuff is observational. There's a couple papers showing like genetic evidence for it on, I think, 3 or 5 species, something like that. And then probably I know of another 3 or 5 that have like papers with observational. I would assume it happens in most of them because they are like sperm cast breeders. But I don't know that we know it for sure happens in every species of Unionid.
Any questions from the room here?
So is the point of the rate of raising them in captivity just to make sure that more of the offspring survive?
Were you able to hear that, Nichelle, and everyone
Yes.
online?
Yeah, I'm just trying to get back to the room and stop sharing my screen so I can see everybody. Yeah, so the point of captive rearing then would be to try to keep as many of those juveniles during those really sensitive ages. Like, so there's a lot of opportunity to lose individuals from the inoculation point through them being, 1 centimeter, 2 centimeters or larger. Those are pretty sensitive times. So the idea is with captive rearing, we would be able to keep more of those individuals.
Dalen has a question.
Nichelle, when you look at the taxonomic variability and the suggestion that, we have more species than we actually have listed, who's going to do this? What's the timeline? And how do you come up with new names?
Right. Yeah. That's a great question. So Dave and I have talked about continuing that work with, you know, new students that he'll bring on to do the morphological assessments to get museum samples, to get wild individuals and do morphometrics to see if we can find out if there is a morphological component to those 4 lineages in occidentalis being distinct. I would be pretty cautious about using just this data to relist everybody. But future work definitely will do that. And I'm excited to be involved in that as Dave moves forward. I hope that answered your question.
Questions online, you can put it in the chat, you can unmute yourself, or
I have one more over here, Dave.
okay, sure.
This is, so with the multiple paternity, is there a skewed sex ratio in the wild? Are there more males than females in general, or is it each?
Okay. Yeah, so Sally asked about sex ratios in the wild, if there's skewed ratios of males and females. And to my knowledge, at least for the populations I used, the sex ratios are pretty close to even, to my knowledge. That could have changed since, you know, I last looked, who knows?
There is a question online. If these splits were to happen, taxonomic splits were to happen, do you think some of the new species would need to be listed under the Endangered Species Act?
Yeah, potentially. And one of the things that I didn't talk about in here, just for time reasons, is there's one region in the Ozarks Wash Hog called the Neosho, which we weren't able to sample. And, you know, that may also be a distinct lineage. So a lot more work is needed here. And I definitely think if we describe those as new species, they will need to be assessed. For whether they need protections or not. But based on other unionids ranges, narrow range endemics tend to need to be listed at some kind of status.
Right.
Hey, Dave. Hey,
no.
Nichelle. This is Nate Johnson. I had a quick question, follow up.
Yeah.
This was a great presentation. I really learned a lot and enjoyed that. Awesome work. So sometimes we wrestle with trying to figure out if we're looking at distinct species or population structure. And some of the models that we use, like SNAP, have been kind of criticized as showing, you know, population structure. You mentioned follow-up work with the morphometrics, which I think is great. How did you kind of gain confidence that you're looking at distinct species and not just allopatric populations?
Right. Yeah, that's a great question. I kind of used the bar as these 4 lineages in occidentalis were so much more different than the 2, you know, population structures in Fasciolaris, right? Like those come out as 2 distinct populations with cluster analyses, but occidentalis come out as much more different. So that gives me a little more confidence, but like we can continue to analyze and do more. And I think we should, like you mentioned, I think morphological assessments will be really important. But since they are declining so fast, like we'd rather be on the side of being overprotective than under. So that's kind of where I fall, but there's definitely room for debate in there.
Good answer. Thanks.
All right, I think we're gonna get ready to move on to the closed defense section. For committee members who are online, you have a separate link that you'll join after this. We'll probably take 5 minutes or so to use the restroom and grab a donut in here for Derek and I who are sticking around. We got a lot of donuts, so we're not eating them all. Thank you everyone for joining us, and excellent presentation, Nichelle. We're all really proud of you.
Thank you. Thanks everyone so much. Bye everyone.
See you on the flip side.
See you in a couple minutes.
Send her a message, but yeah, if you would pass on. Not only was it just